effects, okay? So pleiotropy, you have several genes, and you have one gene contributing, it's the reverse of polygenetics, contributing to many different phenotypic effects. So your book talks about the sickle cell anemia. There's several diseases. Tay-Sachs disease, we have cystic fibrosis is one, where you're going to have a lot of diseases. You'll have a lot of different symptoms, okay? But it's just one gene that actually contributes to it, okay? So sickle cell anemia and cystic fibrosis are both examples of that. Sickle cell anemia is in red and bolded, so I'm trying to really tell you that you need to know that example. Not sickle cell anemia, cystic fibrosis. I apologize. I think I'm getting tired. Cystic fibrosis. Both of these, though, on the worksheet that you have to fill out for me, you need to know both of these and how they work. And your book does a wonderful job of describing that. Page 277, it goes into quite a lot of detail of what happens with these diseases. So sickle cell anemia is where you will have it affects the red blood cell and the shape of it. This is showing you right here where you have, this is your normal red blood cell, and this is the sickle shape of red blood cells. Cystic fibrosis, on the other hand, is where you're going to have the pleiotropy effect of the lungs and the pancreas and the digestive system all starting to fail with those. So let's look, and it's usually, again, this is where you have the normal allele for cystic fibrosis codes for basically that membrane protein that functions in the transport of chloride ions between certain cells and the extracellular fluid. When those chloride ion channels are defective, which is where you would have cystic fibrosis, you're going to have two recessive alleles. They have high concentrations of chloride in the extracellular matrix of the cell. And these are the babies that they kind of claim will taste salty. You guys kind of laughed at that when we talked about that. But basically, this mucus that builds up creates a bunch of problems, and it affects the lungs, the pancreas, and the digestive system. This is showing you, this picture here is showing you sickle cell and how it causes, again, to express sickle cell, you have to have two copies. And these are the array or the pleiotropy of effects, okay? You're going to have the breakdown of red blood cells, the clumping of cells and clogging of small blood vessels, and an accumulation of sickle cells in the spleen. And then you have this cascade of events and basically phenotypic effects, okay? You're going to have an impaired mental function, paralysis, pneumonia, rheumatism, which is basically like your joints hurt, and kidney failure. So heart failure, anemia. So these are all these various phenotypic effects from sickle cell anemia. Okay, so let's look at these table talks. A scientist discovers a DNA-based test for one allele of a particular gene. This and only this allele, if homozygous, produces an effect that results in the death at or about the time of birth. Of the following, which is the best use of this discovery? If you'd like to figure this out on your own, go ahead and pause. And I'm going to go ahead and give you the answer now. So screen all newborns of an at-risk population. Well, it's too late then, okay? Because the newborns, it usually results in death at or around the time of birth. So that's too late. Design a test for identifying heterozygote carriers. Well, only the homozygote recessive will actually have this disease. So if we could get the carriers, then if we could design that test, if you knew you were a carrier, you would know then that you needed to make sure that your mate was not a carrier as well. Introduce a normal allele into deficient newborns. That is an extremely difficult procedure. And honestly, right now, we just haven't figured out how to do this. When we can do this, we could cure a lot of diseases. Follow the segregation of the allele during meiosis. That's great, but that doesn't really solve our issue. So I believe B would be the best example, and we have been able to do that with a lot of recessive traits where the carrier does not express it. We go ahead and we will test, and if we know it's a carrier, then we will test whatever we are, I'm thinking of animals, whatever we're breeding the animal with, to make sure that the mate is not also a carrier. Cystic fibrosis affects now we're down here to the second one, the lungs, the pancreas, the digestive system, and other organs, resulting in symptoms ranging from breathing difficulties to recurrent infections. Which of the following terms best describes this? Well, I did not list the following terms. They're going to be listed on your test, but they're not listed here. Well, I believe this is a wonderful example of pleiotropy, where you have one gene. Remember, this is the allele for the chloride ion channels working or not working, and you have to have two recessive. So basically, this is a wonderful example where you have two genes having multiple effects, so pleiotropy. Which of the following is an example of polygenetic inheritance? Pink flowers and snapdragons. Now let's again go back to what polygenetic inheritance is. It's basically many genes contributing to one phenotypic effect, okay? So if we look through here, our only example would be skin pigmentation in humans, okay? All right, sex-linked traits I'm going to go into very briefly. These are located on the sex chromosomes. That's why we call them sex-linked. They're either on the X or the Y chromosomes. And again, remember, this should be a review. The genotype for females is XX, and the genotype for males is XY. Okay, many sex-linked traits are carried on the X chromosome. And that's why a lot of times sex-linked traits that are on the X chromosome, we will see that males have an increased expression of sex-linked traits that are recessive that are on the X chromosome, because males don't have a spare copy. So the X chromosome that they get and the gene that is on that X chromosome is all they have. Whereas females, we have a spare tire. See, because we have two copies of an X. So the two copies means that if we get one faulty gene on one X, we've got another X, that spare tire that might save us in the long run. Okay, so an example of sex-linked traits would be the eye color in fruit flies. And you're going to examine fruit flies in your lab. And the eye color in fruit flies is only found on the X chromosome. Okay, so if you had a white-eyed female, then that means she has to have two copies because it's recessive. She's got to have two copies. If you had a red-eyed male, he only has the regular big R, okay? Now, if we cross these two, we would know it was sex-linked if all the males ended up with white eyes. Why? Because the